Now let us discuss how the genetic material it is transformed between the cells. Three methods are there. One is transformation, second is conjugation, and third is transduction. So there are three types of gene transfer in the bacteria. In transformation, there is uptake of free DNA. In conjugation, there is direct transfer of DNA from one bacteria to the other bacteria. Whereas in the transduction, there is transfer of bacterial DNA by the bacteriophage. So let us discuss them one by one. The first is the transformation. So before going to the transformation, you should know that the Griffith, he conducted an experiment in 1928. So Griffith was studying a type of bacteria that is known as S. pneumoniae, which can cause pneumonia in the humans. So he noticed that there were two forms of this bacteria. The one was smooth form, that is the S form, F strain, and other was the rough strain. The smooth strain had a protected outer coat. So the smooth has a protective outer coat that make them harmful and deadly to the mice. Whereas the rough strain lacked the protective capsule and therefore they were harmless. Now Griffith want to know that why the F strain was deadly to mice while R strain was not. So he conducted a series of experiments with mice and these two types of bacteria. So in experiment 1, he injected mice with live F strain. So F strain was harmful. So on injecting, the mice become sick and died because F strain was deadly. Now F strain was harmful. So the mice died. Now in experiment 2, he injected mice with live R. The mice remain healthy because R strain was harmless. So mice remained healthy. Now in experiment 3, the Griffith killed the F bacteria by heating them and hence making them non-viable. He then injected these heat-killed S bacteria into the mice. So the mouse here live healthy and the mice did not die. In the final part of experiment, Griffith mixed the living R strain and the dead F strain and injected this mixture into the mice. He was surprised that the mice here got sick and died. However, with giving individually living R strain, in both these, the mice lived. So, but on mixing living R and dead S strain, the mice died. So Griffith experiment was first evidence of what we now call as bacterial transformation. It showed that the genetic material could be transformed between the bacteria, and this transfer was responsible for transformation of R strain into the F strain. So this work laid the foundation for understanding that DNA as a genetic material. So transformation, it is one of the main mechanism of the gene transfer in the bacteria. So it is a process by which bacteria can take up and incorporate exogenous DNA, that is the external DNA from the surrounding environment into own genome. So this DNA can be in form of free floating fragments or even released from the live bacteria. Now in the transformation process, bacteria have developed specific mechanism to take up DNA from the environment. So this can occur when the DNA from the live bacteria or free DNA fragments released into the environment. So the DNA taken up, it is the DNA that is taken, it is fragmented by the it is fragmented by the restriction enzyme. So the DNA taken up, it is fragmented so that bacteria need to prepare it for integration it into own genome. The prepared DNA is then integrated into the recipient bacterial chromosome. So the enzymes that are responsible for these are the restriction enzyme. The first is the restriction enzyme. So think of restriction enzyme as molecular scissors. So what they will do, they will cut the DNA at specific places. In the bacterial transformation, these scissors are used to cut the new DNA we want to put into the bacteria. And also cut the bacteria's own DNA. So this cutting, it is necessary to combine the new DNA with the bacterial DNA. The other enzyme is the DNA ligase. It will act as a glue. So it will act as a glue that stick the cut DNA pieces back together. So after the restriction enzymes cut the DNA ligase, it helps to seal the new DNA into the bacterial DNA, creating one continuous piece of DNA. So once the DNA gets integrated, the new DNA may contain genes that the recipient bacteria can express. So transformation has important implication. The transfers of genes, including those encoding antibiotic resistance via the transformation, it is a critical factor in spread of antibiotic resistance among the bacteria. Transformation, it is also used in laboratory to introduce specific genes. So it can be used in a research tool. In the biotechnology, the transformation, it is used to produce proteins, enzymes, or other products. So it is also used in a biotechnology. So in the bacterial transformation, the first you need a piece of DNA that you want to put into the bacteria. So this DNA usually contain a specific gene or information that you want the bacteria to have. So second step is cutting the DNA. Think of DNA like a long twisty ladder. To fit into the bacteria, we need to cut into specific sports. This is where the restriction enzymes comes in. They act as a scissors, cutting the DNA at specific locations. So now, so after cutting the DNA, it has a ragged edges. So to put it back together, we use is a DNA ligase. So that act as a glue. It helps to stick the DNA back together. Now we need to prepare the bacteria to take into the DNA. So we mix the cut DNA with the competent bacteria. So the bacteria take in DNA from its surroundings. It's like they are eating the DNA and making it a part of their own genetic code. So finally, we give the bacteria a nice home with the nutrients so that they can grow and multiply. As they multiply, they pass on the new DNA to their offspring. So this is bacterial transformation. Other method is transduction. So transduction, it is a process by which the genes they are transferred from one bacteria to another with the help of a bacteriophage. So in this, the bacteriophage would act as a carrier. So the process involves the bacteriophage that will affect the donor bacterium, take a portion of donor DNA with it, and then delivering that DNA into the recipient bacterium. Now these bacteriophages they exhibit two distinct life cycle. One is lytic and other is lysogenic. So these cycles they have a different outcome for the host bacterium and the phage itself. The lytic phage, remember, it is also known as virulent phage. So in the lytic cycle, the bacteriophage is referred as virulent. Its primary goal is to replicate and produce numerous progeny while causing the destruction of host bacterium. So the bacteriophage, here it would come and attach to the surface of the host bacterium. So the phage would, in second stage, the phage would inject its genetic material, usually the DNA, into the host cells. So inside the host cells, the phage would multiply. So inside the host cells, the phage genetic material take over the host machinery for replication and transcription, leading to synthesis of new viral components. So the new phage particles they are assembled from the synthetic component. So the host, the last step is lysis. So the host bacterium, it lyses, it bursts, releasing the newly formed phage particles that can go into the another host bacterium. This one is the lytic phase. Other is the lysogenic phase. It is also known as temperate phase. So in the lysogenic phase, in the cycle, the bacteriophage, it is referred as temperate. So rather than immediately destroying the host cells, the phage integrates. Here, the phage would integrates its genetic material into host bacterial chromosome. So here the phage integrate its genetic material into host bacterial chromosome. So lysogenic cycle, it's proceed as follows. First of all, similar to the lytic phase, the phage would attach to the host bacterium and it will inject its genetic material. So instead of replicating immediately, the phage DNA would integrate into the host bacterium chromosome. That would become prophage. The third is the prophage replication. So the prophage, it replicates as part of host chromosomes during bacterial cell division. So the prophage, it remains in the host bacterium being passed on to the daughter cells. Remember, under certain conditions like exposure to stress, the prophage can be induced to exit the host chromosomes and enter the lytic phase. So the lysogenic bacteria, they will harbor the prophage and they are unharmed by presence of prophage. So the prophage, it can conform new characteristic to bacteria as it can become an additional segment of the bacterial chromosome. So lysogenic conversion, it is a process where the prophage DNA itself carry a new genetic material and influence the bacterium characteristic. It can have a significant importance as observed in case of Corynebacterium diphtheria. So in this bacterium, the lysogenization with specific phase, that is the beta phage, that is phage beta, it can confirm toxigenicity and thus virulence to the bacterium. So removing the phage from a toxigenic strain makes it non-toxic. So prophage, they are the bacteriophage genome that have integrated into the bacterial chromosomes or exist as plasmid within the bacterial cells. Now the transduction, it can be of two types. One is generalized transduction or second is specialized transduction. In generalized transduction, all the fragments of the donor. So in a generalized transduction, any random piece of bacterial DNA can be packed into phage capsid. In the generalized transduction, any random piece of bacterial DNA can be packed into phage capsid. So this process in generalized transduction, any random piece of bacterial DNA can be packed into phage capsid. So this process occurs during the lytic cycle of temperate phase. So this process occurs during the lytic cycle of the temperate phase when the phage infects a bacterium and starts replicating. It can incidentally pack a piece of bacterial DNA instead of its own genome. Whereas in the specialized transduction, only specific genes or the specific region of bacterial DNA can be packed into the phage capsid. Uh, this process occurs during the lysogenic cycle of temperate phage. So when the phage integrates its genome into the bacterial chromosome. So the generalized transduction, it is a random process that can transfer any piece of bacterial DNA, while the specialized transduction, it is a more specific and controlled mechanism that transfers specific genes or region of the bacterial chromosome. Well, the conjugation, it is mechanism of the gene transfer in bacteria when the genetic material it is transferred uh, from the donor bacterium to the recipient bacterium. So there is transfer of the genetic material primarily the plasmid that plays a significant role. So the conjugation involves the transfer of the genetic material, especially the plasmid, from one bacterial cell to the other. So plasmid, we know they are the extra chromosomal circular DNA molecules that can carry various genes, including those for the antibiotic resistance. The bacterial conjugation, it was first discovered by Lederberg and Edward Tatum in year 1946. So they conducted a research on E. coli. So in conjugation, one bacterium acts as a donor which contains a plasmid known factor that is known as the fertility factor. So the F factor, it contains genes responsible for formation of specialized fimbriae or the sex pilus. So the donor cell, it forms a physical connection with the recipient by extending its sex pilus. So making a contact. So there is a formation of so the sex pilus, it connects the donor and the recipient cells. So there is a replication and transfer of F plasmid through this sex pilus. So the F factor, it is referred to as episome because it has ability to exist in two states within the bacterial cells. One is the integrated state. In some cells, the F factor become integrated into the host bacterium chromosome. So in this state, the F factor, it is inserted into the host genome. Free state, in other cells, the F factor remains free as separate genetic element, not integrated into the host chromosome. So when the F factor, it is in integrated state, it forms a specialized form of bacterial cell that is known as HFR, that stands for high frequency recombination. So these have the F factor integrated into the chromosome. So these cells will have ability to transfer the chromosomal genes to the recipient cells with high frequency during the conjugation. What is F prime factor. Occasionally, the F factor, it can revert from the integrated state to the free state. So when this occurs, it may carry it with some adjacent chromosomal gene from the host bacteria. So such F factor that incorporates some of the host chromosome gene, it is known as F prime factor.